So, picking up where we left off the other day, we're ready now to talk about the stomach. Stomach is the place where the food, the bolus, as we'll call it, the flexible ball of food plus the saliva that was mixed up there in our mouth, once swallowed or deglutitioned, I don't know if that's a word, but I just made it up, that bolus will make its way into the stomach. Your stomach is a portion of the digestive tube that will dilate or expand into an elongated pouch-like structure. And that's because on the inside of the stomach, there are these accordion-like folds called gastric folds. They also go by rugae, but they enable the stomach to stretch very significantly. And that's very important because that enables the stomach to hold on to a lot of food. Within the stomach, you find gastric glands that contain ducts that can channel their secretions to the cells along the ducts into the lumen of the stomach. You can see here, these gastric glands are going to open up into the bottom narrow channels called gastric pits, and that's what we see here. Gastric pits are just indentations of the mucosa made up of several different types of cells. Let's talk about those cells. There's two types of mucus cells. They are essentially the same thing in that they produce mucus or secrete mucus. Difference is just really where they're located. The surface ones are closer to the surface, whereas the neck ones are down further into the pit. Okay. Then you've got parietal cells, which secrete hydrochloric acid. HCl is a very, very important chemical, as we'll talk a little bit more about here in a second. Helps us with the digestion of proteins. Parietal cells also secrete intrinsic factor, which is needed for absorbing vitamin B12. Chief cells are going to secrete two very important enzymes. Okay, pepsinogen, which is actually not an enzyme, but it is a precursor to the actual enzyme, which is pepsin, which is the only protein digesting enzyme in the stomach. Okay, and so when we pull up our list, we'll add that to it. It is very active in the very acidic environment of the stomach. Remember, we talked about lingual lipase, also very active in the stomach, whereas salivary amylase is not so active in the stomach. Chief cells also secrete gastric lipase. Along with that lingual lipase, gastric lipase helps with digesting triglycerides, forming fatty acids and monoglycerides, so breaking down the fats into smaller pieces. And then you've got other cells, which include enterochromaffin-like cells or ECL cells that secrete histamine and serotonin. We'll kind of find out what those chemicals do here in a little bit. G cells, which you can see down there in the bottom right, shown in blue, secrete a hormone gastrin. Gastrin is a hormone, so it gets dumped into the bloodstream. Along with histamine, gastrin helps stimulate the release of hydrochloric acid. So in other words, G cells with the gastrin and the ECL cells with their histamine help activate the parietal cells. Okay. There's also what we call gastric cells that secrete another hormone, ghrelin, into the bloodstream, which is a hormone that promotes the experience or the feeling of being hungry when your stomach's actually empty. Okay. Now, as far as absorption goes, there's only a little bit of absorption that takes place in the stomach because the epithelial cells are impermeable to most materials. So most materials can't pass through the cells and enter the bloodstream. However, mucous cells in the stomach can absorb some water, some ions, some short chain fatty acids, and even some drugs, which include aspirin and alcohol. So it does enable some things to come into the walls and enter the bloodstream. But for most things, what we'll find out is it's not the stomach, but it's the small intestine, and then to a much lesser extent, the large intestine. Let's go ahead and switch over to my document camera here. And we had this list that we started the other day. We're keeping a track on what chemicals help us with digesting carbs, fats, and proteins. We're not going to add anything here for carbs, but we do have a new enzyme to add for fats, which is gastric lipase. Gastric lipase is an important fat digesting enzyme. And then under proteins, we have pepsin. But I'm also going to add, it's not an enzyme, but hydrochloric acid is an important helper with the digestion of proteins because it helps with denaturing them. denaturing proteins. And we'll talk about that here in just a second, a little bit more. Now, several minutes after food enters the stomach, waves of peristalsis pass over it every 15 to 25 seconds. Most of these waves begin at the body, which is the middle part of the stomach. Okay, you can kind of see where that's taking place right here on the top picture. Okay, and then they intensify as they reach the antrum. The antrum is this region here where the stomach tapers to meet the small intestine. Okay. Each peristaltic wave moves the contents forward in a process, and you can see here with the blue lines with the arrows pointing in this direction. We call that propulsion. Okay. At this point, though, the pyloric sphincter, which is the sphincter that regulates the movement of chyme out of the stomach, is pinched shut. So in other words, the stomach is going to push stuff forward, but it's not going to leak out because this is closed. So like a wave hitting, a wall, what's going to happen is there's going to be backflow occurring. Okay. And so what's going to happen is we're going to have propulsion going forward, and we call this backwards movement retropulsion. And what this allows for is the mixing of that food with the gastric juice, all that stuff that was just discussed on the previous page, which forms chyme. Chyme, therefore, is described as a mixture of gastric contents and gastric juice being the stomach juice. And once again, gastric always just refers to stomach. So that's what chyme is. I'll try to avoid calling it food because it's no longer truly food. It's food plus other stuff, and that includes gastric juice. Okay. Now, once the food particles in the chyme are small enough, so in other words, once we've broken them down significantly, they can pass through that pyloric sphincter, a phenomenon known as gastric emptying. And we can see that here with the bottom figure. It's a very slow process. Only about 3 mils of chyme move through it at a time. And this takes about 2 to 6 hours. So for people that, eat a meal and then 20, 30 minutes later, they rush to the bathroom, they might say, oh man, that rushed through me or ran right through me. They are absolutely incorrect in that statement, because you can't digest your food that quickly, pass it through all the segments of the GI tract. In fact, it's probably still in the stomach, like 99% or more at that point, which means what's being pushed out, what they're going to the bathroom to eliminate, is something they ate much earlier in the day or even the day before. And we eject chyme approximately every 20 seconds into the duodenum, and this emptying of the stomach is controlled both hormonally and via nervous mechanisms. We'll talk a little bit more about that later in the lecture. Let's talk a little bit more about the secretion of hydrochloric acid and the importance of that hydrochloric acid for digestive purposes. During the time when chyme is mixed, digestion by salivary amylase continues until the acidity of the gastric juice inactivates it. Talked about that the other day. Remember, salivary amylase works really well in our mouth, but once exposed to the hydrochloric acid in the stomach juice, it breaks down, it denatures, it loses shape, it loses function. Okay, that acidity at the same time does activate the action of the lingual lipase enzymes, which help begin the digestion of triglycerides. Okay, this hydrochloric acid, as we just learned, is secreted out of parietal cells into the lumen of the stomach. And it is done so via what we call a hydrogen potassium ATPase pump. It is an ATPase pump, which is telling you right away that ATP is necessary to power this process. So it's a form of active transport. We don't truly secrete hydrogen ion or hydrochloric acid. What we do is we secrete the 2 ions, hydrogen ions and chloride ions, which then, when out in the stomach's lumen, come together, and that's what forms the hydrochloric acid. So we're releasing the ingredients, and then they come together in the lumen. Okay, the hydrogen ion gets pumped out through that hydrogen ion, potassium ion ATPase pump. The chloride ions are going out on a different part of the cell through different types of channels, okay, and into the lumen through what we call potassium channels, or excuse me, chloride channels. Okay. Potassium's also moving through, although this picture is not showing it. Okay. But what happens once that hydrogen ion and chloride ion get out into the lumen? Remember, opposites attract, so the positively charged hydrogen ions and the negatively charged chloride ions come together, and that's what forms the hydrochloric acid, which creates the very acidic nature of our gastric juice. Okay. Now, where does this hydrogen ion come from? Where do these hydrogen ions come from? Well, it's actually a reaction that we talked a lot about the other day. The parietal cells that are in our stomach have the same enzyme, carbonic anhydrase, that our red blood cells have, which takes water and carbon dioxide and combines them to form the acid, carbonic acid, which then breaks free to form hydrogen ion and bicarbonate ions. The bicarb, as you can see, exits the cell, eventually making its way into the bloodstream. And while that's moving out of the cell, the chloride is coming in from the interstitial fluid. And so that's where the chloride comes from. Okay, so the parietal cells actually make the hydrogen ions. They don't make the chloride ions, but then they do move them out into the lumen of the stomach. Okay. Now, what is important about hydrochloric acid aside from activating lingual lipase? It helps with denaturing proteins. Remember, denaturing means that, like the slinky, if it loses its shape, it loses its function. What does that have anything to do with digestion? Well, proteins that are denatured are more easily digestible. So that's why I added HCl to the list, because by denaturing the proteins, unwinding them, then, for example, pepsin can more easily digest it. Okay. Also, once again, acidity of the stomach helps activate the enzyme pepsin due to its optimal need for an acidic environment. And as we've talked about before, the acidic environment helps kill many microbes, and we talked about that in our previous unit when we talked about immunity. Okay. Now, the secretion of hydrochloric acid is regulated by kind of three different things. Okay, acetylcholine release from parasympathetic neurons. There are acetylcholine receptors on those parietal cells, and when acetylcholine is released from those parasympathetic neurons, that can activate the secretion of hydrochloric acid. Gastrin, which we just learned, is secreted by G cells. G cells actually part of the stomach. That gastrin is going to bind to gastrin receptors, triggering the same thing, as well as histamine, which is released by mast cells in the lamina propria of the GI tract. So those different chemicals all end up promoting hydrochloric acid secretion. Okay, moving on from the stomach. If we were to follow the tract, we would be going directly to now talk about not the pancreas, but instead the small intestine. But we're going to take a detour here for the next couple slides by talking about three accessory organs: the pancreas, the liver, and then we'll get into the gallbladder. And that's because they collectively produce fluids that get dumped into the small intestine. Okay. So then once we talk about those three organs, then we'll get back to talking about the small intestine. Okay. We've talked about the pancreas before. Back when we talked about the endocrine system, we learned about what we call pancreatic islets, or islets of the Langerhans cells, which are endocrine in function. They're only 1% of the cells of the pancreas, and they are the cells that secrete hormones that help maintain blood sugar levels or blood glucose levels. The hormones include insulin, glucagon, pancreatic polypeptide, and somatostatin. Mainly kind of focus in on insulin and glucagon because those are ones that really regulate blood sugar levels. The other 99% of the cells are center cells and are exocrine in function. And they are exocrine in function by way of secreting pancreatic juice, which is a clear colorless liquid consisting mostly of water, but also some salts, sodium bicarbonate, and several enzymes. Okay, and those enzymes are all listed right here, which play a very important role in digestion. Now, as far as the pancreas goes, it does not do any digestion. It's not part of the tract. This pancreatic juice gets funneled into the tract through some various pancreatic ducts. There's an actual duct named the pancreatic duct. That's the major duct carrying this pancreatic juice. There's also an accessory duct, but it doesn't really matter. Both of these ducts lead to the same place, which is the duodenum, the first segment of the small intestine. Okay. The enzymes include the following: pancreatic amylase. This is going to be similar to salivary amylase in that it helps with digesting starches. So in other words, we're digesting polysaccharides or carbohydrates. Trypsin is a digesting or a protein digesting enzyme. So it's like pepsin that we just talked about. Pancreatic lipase is the principal triglyceride digesting enzyme in adults. So this is our most important fat digesting enzyme. Phospholipase is not quite as important as pancreatic lipase in digesting lipids, and that's because phospholipase digests phospholipids. Phospholipids are not the major type of fats in our diet. Triglycerides are, but looking at my list, I have them listed just as carbs, fats, and proteins. I don't really specify what type of fats, so I will add phospholipase under that category. And then I'm not going to add ribonuclease and deoxyribonuclease because they don't digest carbs, proteins, or fats. They digest nucleic acids. Nucleic acids, remember, are DNA and RNA. If you don't remember, DNA is short for deoxyribonucleic acid. Well, that's what this digests. Ribonuclease digests RNA, ribonucleic acid. So those are not going to get added. And that's because DNA and RNA aren't fuel nutrients, and they're not a huge component of our diet. So adding in, we have under carbs, pancreatic amylase. Under fats, we've got the most important fat digesting enzyme, pancreatic lipase. I'm also going to add phospholipase, once again, because it's not specifying which type of fats we're talking about. And then under proteins, there's going to be trypsin. Now, there's other enzymes actually that your pancreas creates. There's actually like what we call chymotrypsin. There's a couple others that I'm just going to keep it just to a few, not all of them. Now, the stimulation of pancreatic juice release is controlled by hormones once chyme or food from the stomach begins emptying into the small intestine. So in other words, when the small intestine starts receiving that chyme as the stomach empties, that's when the pancreas starts dumping its secretions into the duodenum. Okay. Triggers for this include a drop of the pH inside the duodenum below 4.5. Okay, this causes the release of secretin, but notice who is secreting secretin. It's not the pancreas, despite the fact that we're talking about it on this page, but secretin is released by glands of the small intestine. So you might want to highlight or underline that. Okay, this hormone stimulates the release of bicarbonate ions, which are the same ions we talked a lot about the other day. Bicarbonate ions are very important because this helps neutralize the acidity of the chyme in the duodenum. Okay. I didn't mention it, but your stomach can handle a very, very low pH. It can handle a lot of acid, and it's because of the mucus that's on the inside of your stomach. It's very thick, and it protects the stomach wall. Okay. Neither your esophagus, which we talked about the other day with like reflux disease, nor your small intestine can handle that same acidity. So what we do is we release a base, which is what bicarbonate is, to essentially just counteract the hydrochloric acid. And so those neutralize each other out, and therefore the small intestine doesn't get, eaten alive by the hydrochloric acid. Okay. Another trigger for release of pancreatic juices is the fat and protein content of the chyme, which signals the release of the hormone cholecystokinin, which, just like secretin, is also released by the small intestine. CCK, as it's abbreviated, stimulates the pancreas to produce and secrete digestive enzymes like those that I just talked about, lipase, amylase, trypsin. Okay. So cholecystokinin, released by the small intestine, tells the pancreas to release these different enzymes into the pancreatic juice. Okay. Secretin tells the pancreatic juice to also contain bicarbonate, Okay. They also will help stimulate the liver to produce bile, which we'll talk here about bile in just a second. Produced by liver and stored in the gallbladder. And that's also going to be something that secretin and CCK stimulate, which is the gallbladder, and trigger it to contract, which is where we store a lot of our bile in order for that bile to be ejected in the duodenum. As we'll find out, the action of bile and the release of these hormones, which triggers the release of bile, helps with digesting fats and oils. Major nerves innervating the pancreas are the vagus nerves, providing parasympathetic innervation. As we know, that's the rest and digest, whereas if there's a fight or flight sympathetic activity going on, that's going to inhibit the secretion of digestive juices, allowing for blood supply to reroute to tissues that need it the most. For example, our skeletal muscle as they exercise. Okay, our next accessory organ, I've already started to mention a little bit about it, is the liver, the largest gland in the body. Divided into lobes and lobules, which are just smaller subunits of it. Okay. We've talked a little bit about this, but not a whole lot. Vascularization to the liver includes arterial blood from a hepatic artery, and then venous blood via the hepatic portal vein, which is a vein that has already received blood from things like your small intestine, your large intestine, your stomach, and therefore contains many of the products of digestion that have been absorbed in those organs. And when I say products of digestion, I'm talking about things that we've absorbed, like carbohydrates and vitamins and minerals and water and whatnot. And we deliver them directly to the liver before we have to deliver them anywhere else. That is because, and I'm just going to jump ahead, in your liver, we metabolize those three fuel nutrients: carbohydrate metabolism, lipid metabolism, and protein metabolism. All takes place in the liver. Okay. Another function, and I want to start by talking about this a little bit more, is the secretion of about a liter of bile a day from the hepatocytes, or the liver cells. Bile is a yellow, brownish, or olive green liquid, having a slightly basic pH, 7.6 to 8.6. And inside that bile, it's mostly water, but you also find bile salts, cholesterol, lecithin, bile pigments, and several ions. It's partially an excretory product, so part of it's going to become just part of our feces, but part of it is a digestive secretion. So part of it's going to help with digestion, and it does so using the bile salts to play a role in emulsification. Emulsification is a form of mechanical digestion, physical breakdown from something big to something smaller. And what we're breaking down are large lipid globules into smaller lipid globules. And if I just jump ahead a couple slides, this is going to illustrate what those bile salts do. Okay. If you think about what we know about fats, from way back at the beginning of the semester, we learned that most fats are hydrophobic, so they fear water, right? Well, that means if you ingest any form of fat, fat's typically just going to kind of bunch together, and it's going to create big balls of fat. And what you don't want to digest are big balls of anything, because it's harder to digest them. Okay. But what we can do is, using those bile salts from the bile, we can essentially chop that big fat glob into smaller ones. And thus that makes them a lot more easily digested. And that's what emulsification is. So I'm going to add under the list of fats, bile. And I'm going to put in parentheses that it emulsifies. Principal biopigment is bilirubin, which we've talked about before, formed from the recycled parts of hemoglobin. And we learned a little bit about that, and how, for example, like strocobilin and urobilin give the color of brown, strocobilin for feces, and urobilin the color yellow for urine. This is all intertwined with recycling hemoglobin, which we discussed not too long ago. In addition to secreting bile, the liver performs many other functions. As I've mentioned, it metabolizes our three fuel nutrients. With regards to carbs, it can help maintain normal blood glucose levels by way of either storing it or releasing it. When our blood glucose levels are low, our liver can break down the glycogen that it stores. Glycogen is just a stored form of glucose. That forms glucose molecules that can be then released into the bloodstream. We call that glycogenolysis. We can also convert amino acids, lactic acid, and other sugars like fructose and galactose into glucose. We call that gluconeogenesis. When our blood glucose levels are high, we can convert glucose into glycogen and triglycerides for storage. We call those processes glycogenesis and lipogenesis. We'll talk about all those big words in our next lecture some more. We've already actually talked about them before. Back when we discussed the endocrine system, those were 6 big buzzwords I defined and talked a lot about. With regards to lipids, we can store triglycerides in those hepatocytes. We can also break down fatty acids to generate ATP. We can synthesize what we call lipoproteins, which are just proteins attached to a fat, synthesize cholesterol, and use cholesterol to make bile salts. We'll talk more about this in our next lecture, but proteins can deaminate, which means to remove the amino group from amino acids, which then enables them to be used for ATP production or be converted to carbs or fats. As a result of that process, so that would result in the formation of a toxic chemical called ammonia, but our liver goes ahead and converts that to urea, which is much less toxic, and we just naturally excrete that out in our urine, which we'll talk more about that here in a couple of class periods when we talk about forming urine. We actually also, and we've talked about this before, synthesize most of our plasma proteins. So things like albumin and fibrinogen, for example, that we talked about just a few weeks ago, those are formed in the liver cells. We process our drugs and hormones that are in the body. Our liver can detoxify substances such as alcohol, excrete drugs into bile. That explains why excessively consuming alcohol, like a habitual drinker, habitual drug user, at times can, over years and years and years, destroy their liver, causing what's called cirrhosis. It just means that their liver is just worn out and overworked. We can also use our liver to alter or excrete the thyroid and some steroid hormones, which is kind of cool. Already kind of mentioned this on the previous page, but we excrete bilirubin, formed from the breakdown of hemoglobin, and secrete that into bile. Synthesis of bile salts, which are used for the emulsification and absorption of lipids, as I've already mentioned. We store things like vitamins and minerals. Special cells in our liver can phagocytize aged red blood cells, white blood cells, and some bacteria. That would be the macrophages that we've been talking a lot about here the last couple of weeks. And then activate the active form of vitamin D, along with our skin and kidneys. Where do we get vitamin D? We know. Aside from your diet, like you can get vitamin D milk, vitamin D comes for free out of the sky from the sun, which is kind of cool. All right, very closely associated with the liver is the gallbladder. And I say very closely associated with it, and that's because it is connected to the liver and is on the kind of backside and bottom of it in a small depression. It is just a sac that stores and concentrates our bile that the liver actually produces until it's needed in our small intestine. Okay, this bile comes to the gallbladder by way of the bile ducts, hepatic ducts, and cystic ducts. And then when the bile needs to be released, it can contract its smooth muscle to eject the bile, the contents, into the cystic duct and then into the common bile duct to be transported to the duodenum, which is in the small intestine. Okay. So, in other words, your gallbladder just is a stored sac, and that's a reason why it's fairly commonly removed. People a lot of times can get gallstones where that bile kind of crystallizes and be very painful. Gallbladders can kind of wear out. I talk about this every semester, and typically I have a student that say, yeah, I had mine taken out a year or two later. People without their gallbladder are still going to produce their bile. It's not the gallbladder producing it, it's just storing it. The liver is who produces it, so they're going to be fine for the most part. they might have to alter their diet a little bit, especially avoiding a super, fatty type meal, because they just won't have that stockpile of bile to release and help with digestion. So looking at this illustration, or the couple of illustrations here on the next page, just shows the connections with the gallbladder, the liver, the pancreas, and the small intestine, how all those fluids that they create or store, in the case of the gallbladder, eventually make their way to the small intestine. And that's why I wanted to talk about these three organs before we talked about the small intestine, because along with the chyme coming from the stomach, these chemicals from the pancreas, from the liver, the gallbladder are all making their way into the duodenum, which is the first segment of the small intestine. And the small intestine is where the true magic happens with regards to digestion and absorption. And I'll explain what I mean by that here in a second. Okay, when I say that most of the magic happens with regards to digestion and absorption here in the small intestines, because this is where it's for the most part taking place. Okay, and a big reason why is just because the small intestine is super long. It's not named for its length. It's about 10 feet long, in fact. It's named because it's a pretty skinny tube. Okay, so its length alone provides a large surface area for digestion and absorption, but that area is further increased by three special features: circular folds or plicae, villi, and microvilli. And they increase the surface area of the small intestine by a factor of 600. So by having these three things, there is 600 more times the surface area that it would normally have if it didn't, which adds up to about 200 square meters, which is about the same area as that of a tennis court. So the surface of a tennis court is essentially the amount of surface area that you have in your small intestines. That's an incredible, incredible amount. And that's why we were able to perform a lot of digestion and absorption. Okay. Well, if I kind of jump ahead, we can look at some pictures here on the next page, kind of explain what those structures are. If you open up the small intestine, what you're going to be able to see, and you wouldn't need a microscope to do it, are these fold-like structures called circular folds. Circular folds also go by plicae. Okay, and there are folds of the mucosa and submucosa about 10 millimeters in length. So obviously 10 millimeters, that's about a third of an inch, and that it would be obviously visible with the naked eye. Okay. Now, we can zoom in here at a couple of those circular folds, and if you look hard enough, you can kind of see it looks like a little bit of like stuff is sticking off of them. Each of those little projections, and if we zoom in here, we can see that those are little finger-like structures called villi. Okay, the plural form of that word villi is villi, and the singular form of that word is villus, which you see here. Okay, the villi are home to some very important cells. I'll talk about those here in a second. But on the inside of those villi are a network of blood vessels, an arterial bringing blood in, a venual bringing blood out, and then in between them is a capillary network. This is where most of the stuff that enters the body through the walls of the gastrointestinal tract are going to end up. So for example, when we absorb carbohydrates and water and amino acids and vitamins and minerals, most of the time that stuff's entering right here into the bloodstream. We also see, and you can kind of see them in the color green here, specialized lymphatic capillaries called lacteals. And if you think back to when we talked about the lymphatic system, we learned that one of the functions of the lymphatic system was to absorb dietary lipids and lipid-soluble vitamins. In the case of fats and fat-soluble vitamins, instead of going into the bloodstream, they come into the lymphatic system, into these specialized capillaries called lacteals. And we've already mentioned this, so hopefully you're remembering that. But those are all structures associated with the villi. Okay. Now, those cells that do most of the absorbing literally are called absorptive cells, and they are found kind of throughout the entirety of these little finger-like villi. And if you were to zoom in at one of those cells, you're going to find that facing the intestinal lumen are little finger-like extensions called microvilli, or what we call a brush border, because it looks like a tiny little brush. Okay. And that increases the cell's surface area. And so what you have, if you kind of factor everything up, you've got folds with tiny little finger-like extensions made up of cells with tiny little finger-like extensions. Okay. And those tiny little finger-like extensions on the cells are called microvilli. And once again, those are found on absorptive cells. Okay. Let's go ahead and talk about those absorptive cells. Okay. Aside from absorbing, they also secrete very important enzymes that we'll call brush border enzymes. And we'll talk about those here in just a little bit. But they help with digestion and therefore absorption. You're also going to find goblet cells, and goblet cells are not unique to the small intestine. Anytime you see goblet cells, it's going to be a direct indicator that they secrete mucus. There's other glands and cells. Intestinal glands collectively secrete about 1 to 2 liters of intestinal juice, which is a clear yellow fluid secreted each day. It contains water and mucus, has a slight alkaline pH of 7.6. And along with the pancreatic juice from the pancreas, it provides a liquid medium for absorbing substances from chyme. And the crevices of these intestinal glands are where you find the cells that secrete the following: lysozyme, which we've learned previously, helps kill bacteria. It's released by what we call Paneth cells. CCK cells, which secretes cholecystokinin. That's pretty easy. CCK, we said earlier, was the abbreviation for cholecystokinin. So the cells that release that literally are called CCK cells. And then S cells secrete secretin. And if you think back, we just talked about those two chemicals back here when we talked about the pancreas, because that's what they target and activate. Cholecystokinin activates the release of these enzymes from the pancreas. Secretin is going to help trigger the release of bicarbonate, which, as we already learned, helps neutralize the acidity that is in the chyme. Okay. Now, that's all stuff associated with the mucosa, the inner lining of the small intestine. Okay. The submucosa, which is the layer outside of the mucosa, also contains duodenal or Brunner's glands, which secrete an alkaline mucus that helps, along with that sodium bicarbonate, neutralize gastric acid that's in the chyme. So another way to help protect from, burning holes in the wall of the small intestine. So obviously, the small intestine is very involved with chemical digestion because there's a lot of enzymes, a lot of chemicals, but it's also involved with mechanical digestion. And there's 2 processes involved here: segmentation, which we talked about the other day, and then a special form of peristalsis called the migrating motility complex, or MMC for short. Okay. The function of the segmentation process helps mix the chyme with the digestive juices and brings the particles of food into contact with the mucosa for absorption. It does not, though, result in the push of the contents along the tract, because that occurs through the other form of peristalsis, or the other form of muscle movement, and that's through the migrating motility complex. Once again, this is a special form of peristalsis beginning after most of a meal has been absorbed. Begins in the lower portion of the stomach, so it actually is traced back to the stomach, and pushes chyme forward along a stretch of small intestine before dying out. Okay, the MMC slowly migrates down the small intestine, reaching the end of the ileum in about 90 to 120 minutes, so about an hour to 2 hours. Okay, and then after that, another process begins back up in the stomach, and that's going to result in chyme remaining in the small intestine for about 3 to 5 hours. So at minimum, if you add the 2 hours from the stomach and the 3 hours here, at minimum, it takes 5 hours to get that food from the stomach to the large intestine. We're not even at the end point yet. We're at the small intestine. We still have another intestine to go. Okay. On the other end, if you add 6 hours, because it was 2 to 6 hours in the stomach, 6 hours in the stomach, 5 hours here, that's 11 hours. Once again, demonstrating that you can't defecate out that food you ate 30 minutes after the meal, despite what some people seem to think. Now, before we dive in on the large intestine, I want to talk about the digestive processes, a little bit more of each of our 3 fuel nutrients. And we've been keeping a running tally on that other piece of paper, but I want to kind of talk about them individually for a couple slides. And then we'll actually jump in and talk about absorption, because digestion breaks things down, and then we're able to bring those things in and absorb them. With regards to digestion of carbs, we need to take those carbs from their polysaccharide state. That hopefully is the most common form of polysaccharide, most common form of carbohydrate in our diet. Okay, hopefully we're eating complex carbs and less of the simple sugars. Okay, but those polysaccharides, which are long chains, must be broken down into the monosaccharides, into the individual building blocks. We learned earlier in this lecture, we talked about this the other day, that salivary amylase begins that process until it's inactivated by the acidic pH of our stomach, which means that only a few starches are broken down by the time the chyme leaves the stomach, which then means that pancreatic amylase is going to have to take over a good portion of the job. Okay, pancreatic amylase released into the small intestine helps take those polysaccharides and break them down into disaccharides. And at that point, those disaccharides like sucrose, lactose, and maltose can be broken down by brush border enzymes, which include sucrase, lactase, and maltase. And as a result of this process, we end up forming the individual monosaccharides like glucose and fructose and galactose. And so we've got 3 more things to add to our list under carbohydrates. We have sucrase, which breaks down sucrose, the common substance we commonly call sugar. Maltase, which breaks down maltose, malted sugar. And then lactase, which breaks down lactose. And where do you get lactose? Milk and dairy products. Okay, people with lactose intolerance, I'm sure you've heard of that before, don't produce a good form of lactase. They either don't produce enough or they produce kind of a faulty kind, and that leads to them having a hard time digesting sugary milk. Milk sugar is what lactose is called, or lactose from milk. Now, not all carbohydrates that we ingest are actually digestible. Cellulose is actually completely indigestible. It is not readily broken down. We don't have the capability to do that, to break it down. We commonly call cellulose fiber in our diet. Okay? And this is beneficial. We don't really get any nutrients because we can't break it down from it, but it adds bulk to our diet. So, we're not going to gain weight by eating something purely with cellulose in it, because you can't absorb that into your body. It speeds things up through the digestive tract. If you ever, had a Fiber One bar or something with fiber, or if you like taking Metamucil, you might have experienced a quicker run to the bathroom after putting that in your body. It speeds things up, which actually is beneficial, and reduces risk for things like colon cancer. Any fruit and vegetable type of diet is going to get you lots and lots of fiber, therefore a lot of cellulose. If you want to think about like corn, for lack of a better way to say it, you eat it, you chew it up, swallow it, and then when it gets pushed out, it looks exactly like it did when you brought it in your body. It doesn't get broken down really at all. So once again, digestion of carbs ends with the production of monosaccharides, which then can be absorbed by the small intestinal wall. So that's carbs. Let's talk about proteins. Remember, proteins consist of chains of amino acids. We can see an example of one down there at the bottom. In order to absorb proteins, we actually have to break them down into amino acid form. Begins with pepsin, which is an enzyme of the stomach, helps fragment the proteins into peptides, which are just smaller chains. And then we really amp things up thanks to the pancreatic juice, which contains trypsin, chymotrypsin, carboxypeptidases, and elastase. So you can see several more listed here than what I wrote down. All I had wrote down was trypsin, and that's all that I included on that slide, and that's all that I'm worried about you knowing for the exam from the pancreatic juice. Okay. I am going to add, though, two final enzymes, aminopeptidase and dipeptidase. And so I will add those down here. These are like sucrase, lactase, and maltase. They are brush border enzymes. So they're produced, dipeptidase, produced by those absorptive cells in the small intestine. And they're the ones that kind of complete that digestion. They break down the proteins to the point where we're at individual amino acids, or in some cases, we only end up breaking them down to where there's a dipeptide, 2 amino acids, or tripeptides where there's 3. Now keep in mind, under the list of proteins, I don't have listed here, but it did help play a role in digestion because it helps with activate the pepsin, and that's hydrochloric acid, HCl, which, as we learned, helps denature the proteins, unwinds them so that it makes it easier to digest them. And then at that point, we can absorb the free amino acids into the blood and then eventually carry them to the liver. With regards to fat, it is a little bit more complicated than digesting carbs and digesting proteins. We're going to use enzymes known as lipases. Once again, remember the primary type of primary type or abundant type of lipid in our diet are triglycerides. We can see several of those examples here. Each triglyceride, if you don't remember, is a 3-carbon molecule with 3 fatty acids attached to it. That's where the tri comes from, 3 fatty acid tails. You can see these little lines, and then the glycerol molecule is kind of horizontal bar. Okay. What we need to do is eventually break all that down and apart from one another. Okay. It all begins in the stomach with lingual lipase and gastric lipase, although lingual lipase not produced in the stomach, but produced by our lingual glands on the surface of our tongue, but then doesn't begin digesting until it hits that stomach where the acid activates it. Okay. Those 2 enzymes, lipid and lingual and gastric lipase, begin the digestion of lipids in the stomach. But the true major fat digesting enzyme is pancreatic lipase, which is released from the pancreas into the small intestine. Okay. At this point, we can then break down the fats into fatty acids and into monoglycerides. But before a lipid globule containing triglycerides can be digested in the small intestine, we need to emulsify it, which is where we take those bile salts and we take the big globs of fat and we break them down into smaller fat globules. And we can see that taking place thanks to the bile that's produced by the liver and stored in the gallbladder, which then results in the formation of what we call micelles, which are shown down here at the bottom. And we'll talk a little bit about micelles here in just a little bit. So that's digestion. And at this point, our list is through. And here on this next page, table 25 point whatever, I can't see what the point is, but that's not a big deal, is a listing of all those different enzymes, where they come from, whether it's from saliva, from the stomach, gastric juice, from the pancreas, or from the small intestine and the brush border enzymes. Notice there's quite a few that I did not list off. For example, like I can see that elastase, I never mentioned anything about. I never mentioned anything about enterokinase, alpha dextrinase. And what I listed on the sheet of paper is what you'll need to know for the exam, okay, which includes the accessory digestive chemicals that you don't find here because they're not enzymes. But I want you to make sure you know that bile helps with digesting fats and that hydrochloric acid helps digest proteins. So this table's handy, but you know, the list I gave you and we've been working on is the one that you need to know. Now, once again, the whole reason that we eat food and we drink water is to bring stuff into our body that we cannot produce ourselves. That way we can deliver those things to ourselves after we've done the absorption. And the whole reason we broke all those complex carbs and fats and proteins down is to this point where we can absorb them, the passage of substances through the intestinal mucosa into the blood or into the lymph. Okay. In the case of absorption, it's either going to be through diffusion, facilitated diffusion, so we're going from high to low using a protein. In the case of water, it's going to be osmosis. And in some cases, it's active transport, which means we need energy in the form of ATP to perform it. We'll go through and talk about absorption of carbs, proteins here on this page. On the next page, we'll talk about fats. But those aren't the only things that are part of our diet. We've also got electrolytes. Remember, electrolytes are just like ions, sodium, potassium, chloride, so on and so forth. We've got water, because that's something that we also must absorb a lot of. And then vitamins as well. And there are more other things that I could add. You know, you could add minerals in there, but I just kind of stuck with some of the most important things that our body must absorb. Okay. In the case of carbohydrates, the only carbs that we can absorb are those that are in monosaccharide form. So in the individual building blocks, once again, we've already talked about this, but only indigestible cellulose and fiber would be left in feces. We can't digest those things, so we just poop them out. And when it comes to monosaccharides, we're going to pass that monosaccharide, which there's three types, fructose, glucose, and galactose, either through facilitated diffusion or active transport. In the case of glucose and galactose, they're going through secondary active transport, which means a piggyback ride on the back of sodium. As sodium comes into the cell, glucose and galactose can hitch a ride into the cell. Whereas in the case of fructose, it doesn't need a piggyback ride to get into the cell. Now, once in the absorptive cell, which is what we see here, then those substances can just go through facilitated diffusion, which is just where they use a protein to leave the cell and then go into the bloodstream. Okay. In the case of proteins, or in other words, actually the amino acids, we absorb those in most cases through active transport processes. Okay. And there's different transporters that carry the different types of amino acids. Some carry them in similar ways to glucose. Others carry them with the amino acids by themselves. And we kind of just show those right here in our illustration on the next page. Some active, some facilitated, but we bring them into the cell and then facilitated fusion get in the bloodstream. We can actually absorb di and tripeptides. And it's not quite as perfect, but in the case of dipeptides, we've got two amino acids bonded together. Tripeptides is 3, and they're going to be transported along with hydrogen ions. So they hitch a ride not with sodium, but instead with hydrogen ions. But once inside of the cell, those dis and those tris just get broken apart into single amino acids. So what we end up with, no matter what, if it was a single to begin with or a double or a triple, we end up with the individual amino acids as a result. And then they go through facilitated diffusion to enter the bloodstream. Okay, when it comes to fats, it's all going to be through simple diffusion, which means we don't need something to help carry it. Okay. Now, in the case of fats, it's quite a bit more complicated though. Okay. Remember, as a result of emulsification, which we're kind of showing off right here, those large fat globules end up as smaller fat globules with the bile salts surrounding those. We call those micelles. Okay, here are a couple of those micelles shown. Okay. But inside of those micelles, the triglycerides have been broken into their monoglycerides and fatty acids, the building blocks. Okay. And those fatty acids can either be short chains, so very short, or very long chains. If they're short chains, they can go just directly into the cell and then out of the cell into the lymph. Okay. Or into the bloodstream, sorry. Okay. But in the case of your longer chain fatty acids and monoglycerides, they get brought into the absorptive cells thanks to those micelles, which is like an Uber carrying the fats to the absorptive cells. Okay. Where they then come into the cells, and then we end up reforming the triglycerides. So we break them down and then we build them right back up as soon as we get them into the absorptive cells. Okay. Then we form another Uber structure called a chylomicron. Okay. Chylomicrons then leave the cells by exocytosis. So these are basically just like a membrane surrounding the fats inside. And instead of going into the bloodstream, they make their way into the lymphatic system, into those lacteals. Okay. Now, eventually the lymph, as we know, enters the blood because that's where it all ends up at some point. Okay. About half of the chylomicrons are removed from our blood within 10 minutes after absorption because of the enzyme lipoprotein lipase that breaks down the triglycerides and other lipoproteins into fatty acids and glycerol. We then reform the triglycerides in our liver cells and our fat cells. So it's kind of crazy. We break down the fats, pull them into the cells, we reform them, pull them into the lymph, go into the blood, break them back down, take them to our liver, make fats. So we're just breaking things down, building it back up, breaking things down. So it's kind of just weird. Okay. Looking here on the next page, this is another illustration showing you those different steps where, we talk about carbs and the 3 different types of carbs that we absorb, fats or proteins, excuse me. Here's our fats with our micelles. Here's the chylomicrons. Notice they're going into the lymph where everything else we see goes into the bloodstream. Okay. And once again, this is all taking place in those little finger-like extensions of our small intestine called villi or villus, since we're only showing one. When it comes to electrolytes, I don't have a picture for those, but that's okay. Those are absorbed from the secretions from our various organs so far. For example, as we know, we release hydrogen ions, chloride ions in the stomach. And those aren't the only place we're getting electrolytes. Obviously, we're getting them through our food and our drinks as well. Sodium is absorbed via a sodium-potassium ATPase pump after they have moved into the absorptive cells via diffusion and secondary active transport. Other ions are absorbed via active transport. Most of our water is absorbed in the small intestine via a process known as osmosis. Small amount is absorbed in the large intestine. That's where we kind of finalize it. We'll get to that here in a second. Water reabsorption is dependent upon absorption of electrolytes and nutrients, which helps maintain osmotic balance, just water balance. And then vitamins, if they're water soluble, they go through absorption via simple diffusion. And if they're fat soluble vitamins, they're just like fats and they use micelles. So if you basically just showed right here, fat soluble vitamins instead of these fats, it would be the same thing. As far as the exam, are you going to have to know like all the ins and outs? Am I going to ask you, which ones are going through like facilitated diffusion? You're not going to have to know like fructose, for example, facilitated diffusion. Okay. Know the major takeaways, like, here, know that most proteins are via active transport. Okay. Know that carbs have to be absorbed by monosaccharides. Know that most fats are absorbed using complicated structures called chylomicrons and micelles. So just kind of the overall arching processes, not the specific details. And at this point now, we are finished talking about those last 4 organs. that are very related to one another: pancreas, liver, gallbladder, and then where everything's kind of taking place, the small intestine, which leaves us with one final segment, which is the large intestine. This is the terminal or end portion of our GI tract. Gets its name for its width, not for its length. It's about 3 inches wide, 6 centimeters, and length, I want to say it's like 6 feet, 1.5 to 1.8 meters. There's two main parts to the large intestine: what we call the cecum, which is just the first few inches. It's down there on the bottom right side of your abdomen, and it's kind of like a dead end street going in the wrong direction, because where that chyme, once leaving the small intestine, needs to go is actually up and into the colon, which is the remaining length. It includes an ascending, a transverse, a descending, a sigmoid, and the rectal portions. And this is where we essentially complete the absorption of water, a little bit of absorption of some other stuff. We actually produce some vitamins in here. We form feces, and then using some processes, we expel the feces from the body. Talked a little bit about this morning in anatomy, but I didn't really get into it in too great a detail. But within the large intestine is a large population of bacteria. We collectively call them our intestinal microbiota, okay? And essentially our intestinal bacteria, which provide us, their host, with several benefits. It's a very nice mutualistic relationship. They benefit and we benefit. It's very nice in how it all works. Okay. What they do for us is they produce vitamin K and folic acid, which are absorbed across the wall of the large intestine. They produce short chain fatty acids, which can be used by the cells of the colon for energy and can aid in absorption of different materials. And they can reduce the ability of pathogenic bacteria to cause disease by promoting anti-inflammatory processes. Okay. So that's what they do for us. And they also, and we'll talk about that here in a second, help us with chemical digestion. Let's first talk about the other form of digestion in the colon, which is mechanical digestion. Okay, remember, mechanical digestion is a physical breakdown. In this case, it has a special name. We call it haustral churning. And it's known as haustral churning because the large intestine is made up of these little pouches called haustra. Okay. And what we do is we churn and pass and churn and pass. And the chyme eventually just gets churned and churned and churned all the way to the other end. Okay. So that's what we're doing using those haustra, and that's why we call it haustral churning. Okay. Peristalsis also occurs in the large intestine, but at a slower rate than in the more proximal portions of the tract. The final type of movement is called mass movement, a strong wave of contractions that begin at the middle of the transverse colon and quickly drive the contents of the colon into the rectum. This is similar to peristalsis, except that the contraction lasts for a longer period of time. Usually they take place 3 or 4 times a day, during or immediately after a meal. This is why people have that urge to go to the bathroom after they've eaten. Not because the food that they actually just got done eating has passed through them, but when the stomach fills up with food, this triggers mass movements in our large intestine. We call this the gastrocolic reflex. And so essentially when stomach starts to stretch, it just kind of starts the train moving farther down the line. Now, since these happen several times a day, it might explain, or it does explain why, like if you have that urge to go to the bathroom, but you don't do it immediately, why that sensation kind of can disappear. And that stuff gets driven down in the rectum, which at that point you're going to feel like you need to go. But then when that mass movement is over, things kind of back up a little bit, and then that kind of sensation will disappear, unless it's kind of an emergency, which you don't really have that luxury. Chemical digestion involves mucus secretion by the glands of the large intestine. No enzymes for digestion are secreted here. So once again, our list is over. Okay, we prep the chyme for elimination using the bacteria, which ferment any remaining carbs. And as a result of this, the bacteria release methane. Methane is what contributes to our flatulence, or what we commonly call our farts. And bacteria also aid in breaking down any remaining proteins into amino acids, as well as the decomposition of bilirubin. And that's what gives feces its typical brown color, as we've mentioned several times. Okay. So bacteria help us with several things, but we give them food that they need. It's not like they're eating it, they just utilize it. It's complicated stuff I don't want to talk about in this class. But they get their energy because of the food that we bring in contact with them. And then they help us do different things. So it's a nice plus relationship. Any remaining water that's not absorbed by the small intestine should hopefully be absorbed by the large intestine. And in doing so, that should bring any remaining electrolytes along with them. So to kind of summarize absorption, as you can see, just a little bit takes place in the stomach. Lot takes place in the small intestine. A little bit takes place in the colon. It's mainly water and some enzyme or some electrolytes. Really nothing once we reach the rectum, because at that point we form feces, which needs to be eliminated. Let's talk about that. Just what everybody wants to talk about, pooping. Elimination is the expulsion of feces from the digestive tract. Fancier term for that is defecation. Those mass movements that I was just talking about push the fecal material from the sigmoid colon into the rectum, which causes the rectum to go from kind of a relaxed state to a stretched and full state. Okay. And as the rectum stretches, it activates stretch receptors in its wall. And those stretch receptors initiate what's called a defecation reflex. Defecation reflex is what results in emptying the rectum. So in other words, pushing the feces out. Okay. Here's how it all works. In response to distension of the rectal wall, receptors send sensory input to the sacrospinal cord. So we don't actually send the signal up to the brain. This is an example where the spinal cord gets to be in charge. Okay. As a result, output from motor neurons in the cord travel along parasympathetic nerves back to the descending colon, the sigmoid colon, the rectum, and the anus. Okay. And what results is contraction of their longitudinal muscles, which shortens the rectum, thereby increasing pressure inside. So as the rectum fills, we're going to start pushing more and more in there, and pressure is going to go up and up and up. Okay. Along with that, what takes place is, thanks to the parasympathetic nervous system, we relax or open the internal anal sphincter muscle, okay, which is a involuntarily controlled muscle. When we are in a empty rectal state, the internal anal sphincter is contracted and the external anal sphincter is relaxed. But look what happens here at step 2. Internal anal sphincter is relaxed and the external is contracted. And that's important because the external anal sphincter is voluntarily controlled. This is the one that we have conscious awareness of, just like we have conscious awareness of every other skeletal muscle in our body, right? Well, I can mentally, purposely do what I'm doing with my fingers or move my mouse around. Okay, we have conscious awareness of this external anal sphincter. And at this point, it's going to prevent you from pooping. Okay, because it's not always a good time just to go poop when we feel like we need to, right? Okay. Well, when you want to actually defecate, look what happens. You have to relax both of those muscles to help with pushing stuff out. The muscle wall, or the muscle in the wall of the tract, will contract, as well as your abdominal muscles. We might even perform what's called a Valsalva maneuver, especially if we're going to really have to push. Okay. A Valsalva maneuver involves voluntary contractions of your ab muscles and a forced exhalation against a closed larynx. Okay. So it's like you see it in like little kids. I have a 3-year-old, and you can tell when he was littler and he was still wearing a diaper to poop when he was pooping because his face would turn red. And his mouth was closed. You could tell that he was really pushing. And that's the Valsalva maneuver. Okay. Now, this reflex is one of the very few reflexes that we've talked about all semester long that is able to be suppressed. Only once we gain that ability to hold it. So in other words, when we get potty trained, okay. So a person can postpone defecation by voluntary contracting their external anal sphincter. Feces eventually backs up in the sigmoid colon until the next mass movement stimulates the stretch receptors again, creating the urge to defecate. In infants, the defecation reflex causes automatic emptying of the rectum because voluntary control of the external anal sphincter is not yet developed. Now, we've all probably heard of constipation, diarrhea. Constipation is contents of the lower part of the colon, rectum moving at a slower rate than normal, which results in extra water being absorbed, resulting in a hardened stool. The opposite of that is diarrhea, where the motility of the small and large intestine are increased, causing decreased absorption of water and electrolytes, and thus a watery stool. All right, last slide here is on the kind of summarization of what takes place with regards to digestion and how we regulate it. 3 steps and 3 overlapping phases, because we might have food in one part of the tract and food in another part of the tract. So we're going to experience some of these things taking place at the same time. The first phase is the cephalic phase. The word cephalic always refers to head, and what's up there in our head? Your brain. So this is a reflex involving, or phase involving the brain, regulation by the brain. Okay. The smell, the sight, the thought, or initial taste of food activates this phase. Okay. These neural centers in the cerebral cortex, hypothalamus, and brainstem become activated, which results in activation of the facial, the glossopharyngeal, the vagus nerves. The facial and glossopharyngeal nerves stimulate our salivary glands and start the secretion of saliva. The vagus nerves stimulate the gastric glands to start dumping in gastric juice into the stomach. This purpose is to get the stomach and the mouth ready for the food that it's about to eat. Okay. If any of you guys have dogs, we have 2, and our older one will sit by us on the floor when we're eating dinner, and after dinner, a lot of times I have to clean up his drool, and her drool, I guess I should say. It's her drool. She's not eating food, but she's smelling and she's seeing it. She's releasing lots and lots of saliva, and she's experiencing this. We experience this. Hopefully, we're not to the point where we're drooling on the floor, but even thinking and smelling food, thinking about and smelling food can trigger the cephalic phase. Gastric phase occurs when food arrives in the stomach. Gastric means stomach again. And there's 2 mechanisms involved with this. I already mentioned that earlier, but they're neural and hormonal. Neuroregulation is stimulated when food in the stomach arrives, which activates stretch receptors in the stomach walls, or when pH is changed due to the presence of proteins in the stomach, because the chemoreceptors here detect pH changes. These changes stimulate parasympathetic enteric neurons, resulting in waves of peristalsis and the flow of gastric juice from gastric glands. I talked about that the other day. When the stomach stretches, when the stomach's pH changes, that's when the stomach starts really dumping in its gastric juice, and that's when it really starts contracting and relaxing its muscle and stirring things up. Hormonal regulation is thanks to the hormone gastrin, released in response to distension of the stomach, partially digested proteins in chyme, high pH of chyme due to food in the stomach, caffeine in your chyme, and acetylcholine released from the parasympathetic nervous system. It will stimulate large secretion of gastric juice, strengthen the contractions of the LES, the lower esophageal sphincter, to prevent reflux, so it keeps the stuff in the stomach and not in the esophagus. Increases motility of the stomach and relaxes the pyloric sphincter, which helps promote gastric emptying. The third phase is the intestinal phase, and it begins once food enters the small intestine. In contrast to reflexes initiated during the cephalic and gastric phases that stimulate stomach secretory activity and motility, these ones occurring here have inhibitory effects. Okay, and that results in a slower exit of chyme from the stomach, and I would highlight that. Okay. What this prevents is the duodenum from just receiving all that chyme all at once. Okay, especially if that chyme is super fatty. Okay, because the fat is really hard to digest. Okay, so this results in food staying in the stomach for longer periods of time, which then enables the stomach to digest it more effectively. And then what's already in the small intestine to go ahead and digest what it's got before it receives more. Okay. So the arrival of chyme in the duodenum stimulates sensory neurons that evoke a neural reflex, which inhibits gastric motility and secretion. So when the stomach starts emptying and the small intestine receives that chyme, the small intestine signals the stomach to kind of slow its roll. Chill out, let me do my job before you send me more to do. Okay. Like I said, presence of fat in the chyme also stimulates secretion of these intestinal hormones that inhibit stomach motility, which keeps the fat in the stomach for longer periods of time, allowing the intestine more time to process the fat that it has. All right, any questions?